Hi guys, welcome to the final lecture in biochemistry on biomolecules. Today we're going to talk about the nucleic acids. They're called nucleic acids because they're found in the nucleus of the cell, and they include DNA and RNA, and we'll talk a little bit about our friend ATP as well. So in our last lecture we talked about the central dogma of biology. Of course, a dogma is a tenet or a irrefutable law. And the central dogma of biology is that DNA is transcribed into RNA and then translated into proteins. So our DNA molecule is huge. It's about 3 billion nucleotides in length. And it carries, among other things, codes for proteins. And the human genome was decoded, I think, around 2002. And we've been, at the time they had decoded it, we really didn't know what was there. And so a lot of work's been done in the last 20 years to take a look at that library. You make about 21,500 genes, and then there's all kinds of like regulatory stuff, genetic code that can turn genes on and off or modify other genes. Now there's a bunch of stuff we really don't understand. In fact, the last time I read, about 98% of code or of the strand or the sequences is misunderstood or considered static. So we make about 21,500 different proteins from the 21,500 different genes. And, we can make all kinds of proteins, structural proteins, hormones, carrier molecules, but overwhelmingly, those genes are for enzymes. And the truth is, you've gotten a lot in common with our friend the chimps, right? About 99% homology in our genetic code. But you really have a lot in common with a housefly. And that's because we all are pretty much doing the same metabolic stuff, right? Houseflies eat all kinds of things. And of course, in order for them to digest them, they need the same enzymes. So anyway, DNA and RNA carry this genetic material, and they are polymers of structures called nucleotides. So down here on the left, you see a rapidly cycling image of nucleotides or a nucleotide. And the typical nucleotide is made up of a phosphate group, a sugar, either ribose or deoxyribose, and then finally a nitrogenous base, which is the ring structure here, right? Phosphate group, sugar, nitrogenous base. And in DNA, there's 4 bases: adenine, cytosine, guanine, thymine, and they vary at this nitrogenous base. The, again, phosphate group, sugar, either ribose or deoxyribose. In this case, this is deoxyribose. I should have 2 OHs down here if it was ribose. And then to the right, I have my nitrogenous base. Pardon me. Now, the nitrogenous bases fall into 2 categories. They're either pyrimidines or purines. And, it's a lot of aggravation to draw these 2 ring structures. Ag, A, G. Adenine and guanine are the 2 ring structures, which makes cytosine, uracil, and thymine the 1 ring structures. Now, if you were forced to live underneath the pyramid, when you looked up at night, you might look up into a structure that looked like this. I know you wouldn't, but that's how I remember this. They're the pyrimidines. It's a lot of ag to draw these guys. That makes them purines. Try to commit it to memory. So the nucleotides of DNA and RNA are bound to one another through a specific type of bond called the phosphodiester linkage, right? Notice that it's between the OH group of a sugar and the phosphate group of an adjacent nucleotide. Phosphodiester linkage is achieved through dehydration synthesis, you guessed it. It's a sometimes called the or the phosphate sugar backbone, um, or phosphodiester bond, but phosphodiester linkage is actually the appropriate title. Of course, this is a covalent bond. So in the case of DNA, we have a double-stranded molecule. Two strands. Here's the phosphodiester linkage between two nucleotides here, and we're going to assume that they go off into space in either direction. And then across the way is another strand of nucleotides. Now, these two strands exhibit complementarity. So this strand complements that strand. In fact, the truth is there's always one coding strand and one complementary strand. And what's important about these two strands is they're actually bound together through hydrogen bonds, okay? And so this strand over here complements this strand. And by complementation, we mean that if there's a C here, there'll always be a G here. If there's a here, there'll always be a T there, and vice versa. So these rules, these base pairing rules, are called Chargaff's rules. Okay, so remember, and you can see here that a pyrimidine will always bind to a purine. A pyrimidine to a purine. And importantly, notice the number of hydrogen bonds between C and G is 1, 2, 3, and hydrogen bonds between A and T is just 2. So if you come across some particularly GC-rich regions of DNA, it's hard to get it apart. That's literally some of the chemistry that I worked on when I was at the University of Delaware. Well, DNA is in that beautiful double-stranded molecule, but RNA is not. RNA is a single-stranded molecule. It's RNA because its sugar is ribose. And remember that instead of AGTC, we have AGUC as the 4 bases, right? So it's single strand. AG uracil substitutes for thymine. You'll never see thymine in an RNA. And single strand, the sugar is ribose. Now, RNA looks like the first candidate of nucleic acids on here on planet Earth, because RNA can actually use some enzymatic activity. I know we haven't talked about enzymes in here. I'll probably put that off till later. But there's 3 types of RNA that are important to our discussion in transcription and translation. But there's actually a whole boatload of RNAs. All right, so the 3 types that you might have gone over, or I know you went over in biology, were mRNA for messenger RNA, tRNA for transfer RNA, and rRNA for ribosomal RNA. And though RNA exists as a single-stranded molecule, it can actually fold up on itself into complex structures, just like a protein does. In the case, this is the ribosome here with some tRNAs embedded inside of it. So again, remember that the big differences between DNA and RNA are that DNA is double-stranded. It's CAGT. This is CGAU for ribose. For RNA, RNA is a single strand, sugar is ribose, double strand, sugar is deoxyribose. There's your test question. Last but not least is the nucleotide adenosine triphosphate. All right. So this molecule is the energy money of the cell. This molecule is made by the mitochondria through the process of cellular respiration, specifically glycolysis, and then the Krebs cycle and the electron transport chain. So I make ATP, and every time any useful work is done, I use up ATP. And specifically what I do to ATP is I convert it from ATP, adenosine triphosphate, to ADP, adenosine diphosphate. By breaking that bond there, I release energy to do useful work. Remember, I showed you the kinesin molecule walking down the strand of tubulin every time a step was taken, ATP was used. If I'm a sperm and I want to wiggle my little tail, I phosphorylate or dephosphorylate ATP. Now this doodad here is actually a protein. Number one, you can see its quaternary structure. This protein is the ATP synthase. It's an enzyme. It's a protein complex made up of multiple protein subunits. And the way this thing works is by using a hydrogen ion concentration gradient to spin the pump and generate ATP. So ATP synthase, there's a new enzyme for you to learn about. And I think that'll be enough for today. I'll see you on the other side. Thank you.